Good afternoon. Today it's my pleasure to introduce Professor Catherine Costello, a professor of biochemistry, biophysics, and chemistry at Boston University School of Medicine and Boston University. First, I'd like to say that I was very fortunate to have Kathy as a co-mentor at Boston University, and I can tell you firsthand that it's difficult to find an individual that is more knowledgeable in the area of mass spectrometry and mass spectrometric analysis of biomolecules and the implications of those findings. Kathy received her baccalaureate in chemistry from Emmanuel College, earned her master's and PhD in organic chemistry from Georgetown University, and after over 20 years at MIT, she moved to Boston University School of Medicine in 1994 and established a Center for Biomedical Mass Spectrometry, which includes an NIH NCRR resource and an NHLBI Proteomics Center. Kathy's research interests include determination of structures and functions in biopolymers, with a particular emphasis in glycobiology, post-translational modifications of proteins, and cardiovascular disease and protein folding disorders. And just for a brief aside, to give you a flavor of Kathy's influence in glycoconjugate mass spectrometry, in 1988, she established a systematic nomenclature for carbohydrate fragmentation and spectra of glycoconjugates, and this is still the nomenclature that is widely accepted as the standard. She has co-authored or authored over 280 scientific papers. She's served as president of the American Society of Mass Spectrometry and is the current senior vice president and president-elect for the Human Proteome Organization. She's also the vice president of the International Mass Spectrometry Foundation and a member of the Council of the American Chemical Society and of its boards of U.S. HUPO. Kathy has received many awards, and the most recent was the 2010 ACS Field Franklin Award for Achievements in Mass Spectrometry. So, without further delay, I'd like to introduce Kathy Costello to this fall's lecture. Kathy, welcome.
Thank you very much, John, and thank you and Sandy and Pam and everyone else for my invitation here. It's a great pleasure to be able to come and to talk with you here, and I understand to talk with other people in electronic boxes, and to give you some idea of work that's going on in our laboratory. And what I'm going to be talking to you today about is mostly post-translational modifications of proteins and how these can affect how the proteins associate with one another and with other types of molecules, and affect the outcome of biological systems, to push past the idea of proteomics that mostly aims at just identifying proteins. And what we want to do is to look at complete structures and then look how to these modifications to structures then affect the biological activity. So this, as I picked out the word, is chameleons, is really the basic protein structures show many different faces and activities over time and change back and forth. So as we go from the structures of genes going into proteins, that although there is a lot of variety within the genes, one has to realize as the process of transcription and splicing and post-translational modifications go on, then the number of molecules and different forms of molecules we have to deal with goes up. You notice this is a 2001 reference, so at this point we have 35,000 genes, but I think the relationships are the same no matter what number we pick out. But this makes more and more complex situations and much more challenges for the analytical program, but much better chance to understand the biology behind it. And even as we get within a single set of organisms, like these fellows, the individuals can differ from one another. Within an individual, the number of proteins and their forms within single tissues will be different from one another. Within a single tissue, individual cells will be different from one another, and the proteins within these cells will change with time, and with development, with experience, with environmental issues, and with disease. So we need to identify the proteins. This is representing various forms of glycosylation, disulfide bond formation, and of course, many other changes that are possible. So some of the goals for proteomics and functional genomics is first to determine the total protein repertoire, and this is what's known as the proteome, but then to look at changes in this as a function of these various forms I mentioned, elucidate the post-translational modifications and the resulting effects on the interactions of proteins, to help us develop understanding of the intra- and extracellular pathways, and ultimately to gain understanding of the system, and finally to develop new diagnostics and new therapies finally. The characteristics of mass spectrometer systems are they have now molecular weight ranges to go into the hundreds of kilodaltons, and even megadaltons have been demonstrated. Sensitivity that goes down into the low or sub-picomole range, even to femtomoles, attomoles, zeptomoles. But because mass spectrometers detect all kinds of different molecules, although one can ultimately detect very small amounts of any given material, of course you're going to detect everything else that's present there too. So this very high sensitivity also requires that we work on schemes for purifying the molecules of interest so that they're not overwhelmed by the other materials which are present in the sample, and when we can detect all of these things with this great kind of sensitivity. So it's both a blessing and a curse that we have this sensitivity. We now have fast data acquisition and processing, and this generates very, very large data sets, and we're very dependent on progress in the computational sciences for dealing with these, for processing them and extracting the information out of them. We have to deal with separation methods, both online and offline, and then various kinds of mass spectrometry where we can do the final separation within the mass spectrometer. We have a range of instruments that differ in their resolution. Now I'm talking about mass resolution, so that we could have very high sensitivity with minimal resolution, or a very high resolution with somewhat of a cost in sensitivity, and depending on the project that we're working on, we have to choose the appropriate place in between, and of course also depending on the budget we have, that will have some effect on that. And we can now have mass accuracy, especially in the high performance instruments that are down at the fraction of a part per million, so we can be very specific about the assignments that we're making. I'm going to give, for those who aren't used to mass spectrometry so much, just diagrams of a couple of the major types of instruments. One of the methods we use the most is electrospray ionization. And in that case, a sample in a liquid form in a solvent comes through a capillary, either from a needle or as the effluent from a liquid chromatograph or capillary electrophoresis. There's a high voltage here between this tip and the inlet of the mass spectrometer system. Under these conditions, we form highly charged droplets, and as we go into the vacuum system, pump away the solvent, leaving highly charged sample ions, which can be focused to assure their transfer to the mass analyzer. And then within the mass analyzer, we can separate them based on their mass to charge ratio, and in some types of mass spectrometer, like this ion trap, we can operate it in a selective way that we pick out an ion that we're interested in, kick out the other ones, and further fragment that ion so we can do multiple stages of mass spectrometry. And then in each case detect the ion or the fragments that came from that ion and use that to match to a pattern or to interpret directly to get the assignment of the structure. This type of instrument has very high sensitivity in the tandem mass spectrometry mode, can easily be automated, has a relatively low cost, and moderate resolution and mass accuracy. If we need higher performance, we can go to an instrument such as this quadrupole orthogonal time of flight. Simply means same type of ionization, electrospray, or in this case we're illustrating it also with matrix-assisted laser desorption ionization, where the sample is mixed with a small organic compound that absorbs energy at the wavelength of the laser being used, transfers that energy to the sample to get the sample into the gas phase. Either type of ions are then separated in the quadrupole region and mass analyzed, and we can make it undergo decomposition here in a collision cell where we can put a gas, and then look at either the intact ions or the fragments in the second stage of mass analysis, which is time of flight, in which the ions are accelerated and go through a field-free drift region, here being turned around by an ion mirror to come to a detector. And this gives us a higher resolution and higher mass accuracy assignment of the ions than we got out of the ion trap. So now we have resolutions in the 10 to 15,000 range, where with the ion trap they were, depending on which one you have, from 1,000 to 8 or 10,000. So we have more mass accuracy and easier assignment, but not the opportunity for multiple stages of MSMS. Oops, sorry. If we go to a slightly more complex one that now has an ion trap at the front with all these MSMS possibilities, but low mass accuracy, we can take the ions that go from that into a higher performance stage for detection called the orbitrap. So now we can get the high mass accuracy and high resolution performance in this part of the instrument and still be able to do the multiple stages of MSMS in this part of the instrument. And finally, we can go to the highest performance kind of instruments, which are Fourier transform mass spectrometers. Again, a kind of ion trap, but now within a magnetic field. And we can do such high resolution measurements that we measure to a fraction of a part per million and can get mass resolutions to several 100,000 or even several million, parts of 1 in several million, and have many opportunities for dissociation. With all of them, we can put up the voltage right in the ion source and cause some fragmentation there. In this case, we can also cause fragmentation with a gas collision in the quadrupole region, or the option shown here is to also produce negatively charged ions, have them collide with the analyte in this region, and do what's called electron transfer. So adding electrons to a highly charged species, making a radical product that will then decompose more completely. Or we can let them alone, let them go all the way to the trap, which is in the middle of the magnetic field, and have them interact there with electrons, just electrons themselves, which will cause fragmentation. Or we can introduce photons from an IR laser that will interact either in this cell or in this position, and we can do these steps in sequence. So obviously, the more complex, the price goes up, but the higher performance. So for the ultimate identification, this kind of system would give you the answer, but it's not needed for every type of analysis. In the different types of protein analysis, one would be to take the protein, break it with protease into small pieces, into peptides, separate them by LC, ionize them by electrospray, record the mass spectrum and the tandem mass spectrum, and from that we can get, from the intervals between the peaks and the assignment of the peaks, we can assign the peptide sequence by comparison with a predicted sequence coming from the gene sequence, or by comparison with a library of spectra. The other alternative is to take the protein itself and fragment it in the mass spectrometer, and in that case we generate ions which for each cleavage will give 2 pieces, and the sum of these 2 will sum up to the original mass spectrum molecular weight. And by producing a lot of these, we can get information throughout the sequence. And this works particularly well with the Fourier transform mass spectrometer where now we're producing very high mass ions in many of them. So to get accurate enough assignments and to separate them from one another, we need the high performance instrument. You can think of it this way. When you're doing, looking at a pond, say, and you see an image of something, so you can see a good idea of what your protein looks like and reflect from that up to what it may be. But if you look directly at it, you get a much clearer, much more realistic view of it, and this is more equivalent to what we get with the top-down experiment. In any of these methods, what we're doing for sequencing is to fragment the peptide or protein along a series of bonds within its backbone. Depending on which type of dissociation you use, a different series of ions will be produced. With low-energy collision-induced decomposition, which is the most commonly used technique, what you're breaking is the amide bond here and producing ions called B, which include the N-terminus, and Y, which contain the Y-terminus, contain the C-terminus. If you use these electron-based methods, you will fragment instead at the N-C bond and produce C and Z ions. Either of these can collapse to form these ions which correspond to the A and X series. But in any case, as long as you produce a typical series of the same type of fragmentation, the interval between the peaks will be the weight of single amino acids or an amino acid plus its post-translational modification. So in our laboratory, what we're doing with our mass spectrometry resource and our cardiovascular proteomics center is to develop methods to take samples that come from patients, to use what information we have from clinical background and DNA sequences, combine it with our mass spectrometry to discover what proteins are present and what modifications there are, and other types of studies, particularly in our lab involving the glycobiology part of it. And then we use either 2D gels or multiple stages of LC to separate, to simplify these mixtures, and then different types of mass spectrometry and tandem mass spectrometry to get the information. And as I mentioned, we can select among these different options of mass spectrometry depending on which type of information that we're seeking, how much material we have, what we already know about it. So there's quite a bit of flexibility. If we separate things at the protein level, and we prefer to do this, then we can see the sum of all the post-translational modifications on any single molecule. Whereas if you put it in a gel, digest it, take it back out, and there's multiple modifications, you don't know which ones were on the same molecule, so you don't know the relationship between them. If you separate the proteins and then look at them individually, the different forms of a protein, you'll know which modifications go together. This is looking at different fractions after 2DLC and plotting them out in a heat map form to say what proteins are present. If we take each of those fractions and then do protease digestion to do peptide mass fingerprints, then we can see for each one a different pattern of the peptides and plot it out in a similar way. And this will give us a quick way to look at changes within the samples. So I'm going to illustrate these procedures by a few investigations, and some of them have to do with oxidative stress, and the collaboration is with Martin Feelish, who was at BU but is now in the UK, with cardiovascular disease with Richard Cohen, who's my co-PI on our NHLBI Proteomics Center, and pulmonary hypertension and sickle cell disease with Elizabeth Klings, and in this case a student, Aaron Ohimbino, who is working on this to look in particular at sickle cell disease patients who have pulmonary hypertension. And then in the protein misassembly and misfolding disorders from our amyloid research and treatment and research center, Lorene Corners, David Selden, and Martha Skinner, and our collaborator from Italy, Giampolo Merlini. And I'm going to talk finally about Huntington's disease a bit with our collaborator, Michael Sherman. So first, I said the good, the bad, and the ugly. We'll look at the good. And these are rolls of nitrite. and they're in biology and medicine. And there's been a lot of information that there are biological effects from nitrite, but it wasn't known whether or not these are just temporary or long-term effects. And so what we decided to do was to do a study to give some nitrite treatment and then see what the effects would be on the metabolome and on the proteome. Some of the sources of nitrite are both from our diet and from exercise, inflammation, and from two things that are usually bad-mouthed, but in this case contribute to nitrite, which turns out to be good: air pollution and cigarette smoke, though it doesn't mean I recommend them, and then normal processing of nitrite. So these lead to a transient increase of nitrite in the blood and tissues. So what can happen then is something called ischemic preconditioning, where when there's ischemia, then it's followed by reperfusion, and there's a slow recovery. But if there are brief episodes of ischemia before the real one, then in fact the recovery is more rapid and robust, and there's overall less cellular damage. And the question is, what is going on that would lead to this effect? And so we injected mice with what would be this typical level that you would get from the food or exercise or other sources, allowed the organs to be perfused over a period of 0 to 24 hours, sacrificed the animals, and then looked at their tissues and also did metabolic studies at the same time. 2D page, image analysis to find the spots which were different, excision and identification of those by MALDI-TOF mass spectrometry. And what we could see over the course of 24 hours and even to 48 hours is there are immediate changes in a number of metabolically detectable things and things you can detect, just like nitrosylation, as well as a lot of small molecules, and that a lot of these happen and then go away, but that some changes occur and in fact last for quite a long time. And so we decided to compare the proteomics at the beginning, the proteome, and at this period at 24 hours. If one looks at heat shock proteins, and this is just by Western blot, you can see that there is a dose-dependent effect on them over this period. If we look at the rat heart over this period of time, we found that there were a very large number of spots which changed, and that they fell into a large number of categories of proteins. But there were so many that what we decided to do that was to really subfractionate this and to look in particular at the mitochondria. So we then went back and looked at the mitochondria fraction, and again the control and the 3 different doses, and looked within different groups of proteins to see how they changed as a function of dose at this 24-hour period. And what you can see here is just one of those windows, a single protein with different forms because of post-translational modifications. But if you cut them out and do the protein map, the peptide map, then you get information that by comparison with a database allows you to assign these to aldehyde dehydrogenase, fortunately the mitochondrial form. And if we then look at some of these things and compare them with Western analysis over a function of time, we see with nitrotyrosine modifications that this is also changing, that's dose-dependent and changed over the time and corresponded to what we were seeing by mass spectrometry. We also could do quite accurate quantification of the peptides, and here's one, 2 peaks that are assigned to the same protein, in which we get MS-MS data that gives us complete sequence information from them, and we can see how they're changing. We also see unrelated proteins and look at them over the same period of time. These are the different isotopes from the same protein, a peptide peak, and then these are the peaks at the different dosages. And we can see this curve for concentration for this protein, peptide, and then a quite different pattern for this fatty acid binding protein. And if we go through all the data, we can look at all the different features and their different patterns depending on which kind of map they fall into. And we can see that there are certain types of clusters that are appropriate for the different classes. So they're responding in groups, but with different responses from one group to another. If we look at the ones that fall in this window here, they correspond to what was going on in that Western blot analysis of the glutathione oxidation. And we can then take this data, and one of the staff in our laboratory has written some software that takes the mass spectrometry data and immediately sorts them on the basis of Go functional annotation, so we can look at the biological processes which are affected, the cellular components where they're located, and the molecular functions. So this helps a lot to go through and extract meaning from the data. We also want to go back. I showed you that some of these have different spots attached corresponding to the same protein that correspond to modifications. We can go through, for instance, and look at the phosphorylated forms of the different peptides. And here's two examples where we can, from the MSMS spectrum of a peptide recorded during the LCMS-MS run, find the modification site and locate it specifically to this serine and not either this serine or this threonine. And in the same case, in another peptide from a different protein, we can once again locate specifically the phosphorylation site and get reliable sequence information from the whole peptide to be sure which protein it corresponds to. So in conclusion from this, the single dose of nitrite gave a short spike in the tissue and showed tissue nitrosylation. The transient spike then had major long-lasting alterations in the cardiac redox status and cardiac contractile recovery after global ischemia. I didn't show you these physiological tests or the metabolic ones, but these were consistent with the proteomics result. And the 2D gel-based one and the LCMS-based label-free quantitative analysis detect this initial spike, the initial results, and then these persistent, long-lasting effects and the changes in post-translational modifications, particularly for proteins involved in metabolism and redox regulation. And this gives us some information that these alterations could play a significant role in the myocardial preconditioning phenotype, but of course one needs to study these things in much more detail. Now we get to things which can be bad or sometimes bad, and these are protein variations. And as you're probably aware, with hemoglobins, and this is a mass spectrum, electrospray mass spectrum of hemoglobin, just taken by taking a blood spot, diluting it 1,000-fold, and running electrospray mass spectrometry. You get different charge states. The blue ones correspond here to the alpha chain, and the red ones to the beta chain. With a Fourier transform mass spectrometer, we can resolve all the isotopes of these and get very high mass accuracies, so that we're only off with a few parts per million, even though we're measuring things at about 15,000. But what we've started to do as a possible way of a clinical assay that would be useful for quick screening to find different sequences of hemoglobin that may correspond to diseases, even though many modifications of hemoglobin are not, don't cause problems. Many others do underlie diseases such as sickle cell anemia. Here what we're doing is taking the LTQ Orbitrap mass spectrometer and using the dissociation in the ion source as a first step to produce fragments. Now we know we have both the alpha and beta chains, but we know from looking at them individually what their major fragments are, and we're measuring them with high accuracy. So we can assign the major fragments that come from this nozzle skimmer dissociation, and then select which ones of those we want to do further dissociation to look at sequence variations. But we even know at this point, if these are at the normal positions, they haven't been affected. If they're shifted from the normal position, then they may be one of interest, so we pick that for doing the the further mass spectrometry. So now we've picked the peak that corresponds to the hemoglobin Y96 peak. We see that that's been shifted, and by doing this CID, just collision-induced dissociation, we can get quite a bit of fragment information, and the blue ones are the Y ions, giving us information particularly rich toward the C terminus, maybe not quite so much toward the N terminus, and a few internal ions, but we've written some software now that will help with the assignment of all of these peaks. So that will help us to find differences. And here this is looking at some hemoglobin which had a peak that corresponded by the difference in mass to it being the glutathiolated form. So we can pick that peak here, even if it looks relatively low abundance. We fragment that, and we get not complete sequence, but quite a bit, and enough to tell the difference between the cysteine in this position and the cysteine in this position, because these peaks are not modified by the mass shift of the glutathione, and the peaks which are beyond this cysteine are, whether we read from this direction or read from the other direction. So that enables us to tell, just from this very quick experiment, that the sequence is normal, but in fact the modification is that there's glutathionylation on this cysteine. What we can do now with the FTMS is to do even more elaborate kinds of fragmentation using the two electron-based methods, electron capture dissociation and electron transfer dissociation. Now we select the same peak and do more fragmentation with these methods, and you can see by either of these we get much more complete sequence information, which, even though they're both electron-based techniques, exactly which positions have been fragmented are not the same, so the information is complementary, and we can do them both on the same instrument. If we compare that with, oops, there should have been one more. If we compare that with just the CID spectrum I showed you first, then you can see that, in fact, these techniques are giving us much more complete information. If we look at these patients, then we can look at other proteins and see which modifications are going on. As I mentioned, we're studying pulmonary hypertension in patients with sickle cell disease. So we had one we already could see from the hemoglobin. There was a sickle cell disease. And when we looked at the albumin, now most of the time when you do proteomics, because albumin is the most abundant protein, and there's so much of it that if you don't remove it from the sample, then it's very hard to see the low abundance proteins. That normally we deplete the sample of the albumin and even some of the more high abundance protein, the other high abundance proteins, and then look at the remainder. But we always save that fraction, and we can go back and look at it, because in fact the albumin itself can tell us quite a bit of what's going on by looking at modifications on albumin. So here's the triptych map from the sickle cell patient with pulmonary hypertension. All these peaks can be assigned to the normal peaks in albumin. But these are so reproducible, we can look at these low abundance peaks in the spectrum and compare them from a normal patient to this particular patient here. And what we see is that in this one region, there's similarities here, but there's one set of peaks here that shows up only in the sickle cell patient and the sickle cell patient with pulmonary hypertension, and then another patient with pulmonary hypertension. So this became of interest to see if it was a modification that would say something about the oxidative stress. And in fact, it corresponds to adduction of malondialdehyde at the lysine in position 159, which we can tell by taking this peak and doing MSMS on it. And this is the typical kind of thing where there's an oxidative stress going on, where the lipids are subject to peroxidation, and in the places where there were double bonds, we form aldehydes, and these aldehydes, and particularly 4-hydroxynonenal is one of the most common, can then go and form Schiff bases and other more permanent adducts with the free amino groups on the N-terminus or on the lysines of a protein. And so we can detect this kind of modification by going back and looking at the protein and finding these modifications. If we do LCMS on a digest of the albumin peptides from the same sample and look for the HNE modifications, we see, for example, 2 of the sites correspond to this peak eluding here in the chromatogram and this one eluding in this position, and this shows you the molecular ion regions in their MS spectra. These are then subject to dissociation, and we can sequence them so we can assign the peptide, and by the shift in mass at just this position, we can see that the HNE modification is occurring on this histidine residue. And we go through the entire run in that manner, and we can see from the albumin, we see the modification that I mentioned of the HNE has occurred on all these different sites. The modification of malondialdehyde is on this lysine here. And then there are also glycation modifications, which are occurring, and glycosylation on other sites within the protein. The modifications of cysteine are particularly important, and we've started to call this a thiol proteome. Some of them are important, and they're reversible, and they're needed for a physiological, normal physiology. But as you go to the point where you get more and more oxidation, you finally, in the presence of large amounts of oxidation, reactive oxygen species, then we get pathological and irreversible damage. And we want to be able to probe this, and one of the ways that we thought to look at it is using a technique which originally was developed by Steve Gigi and Rui Ebersold, isotope-coded affinity tags. And they developed it with the idea of using this as a way to quantify proteins in a mix by getting a reagent that would react with cysteines, which are common but not that common, and then they would be able to just attach it to the cysteine, and then, because there's part of the molecule that serves as an affinity tag, after digestion, pull out the modified ones and compare a standard sample with a modified one by using a version of the ICAT reagent, which is stable isotope labeled. And this works quite well for quantifying proteins, but we thought we would use it for another purpose, and that is with the idea that if the cysteine has been modified, it can't react with the reagent, and so it will show up at a lower amount. Even though the protein may be present, that peptide will be depleted. So we can take the normal sample with the unlabeled or carbon-12 ICAT and the oxidant stress sample with the C13 labeled ICAT. As I say, the oxidized forms will not pick up the reagent. We mix the control and the oxidant together, and the ones that have non-reactive cysteine will come out with the same size peaks, and the ones with reactive cysteine, that peptide will show up as though it's lower abundance. If we do that, and here's P21-RAS with glutathione modification, we can see some of them are at the same abundance. Some cases, here's only about a 1/3 depletion. In this case, there's about a 50% depletion. And we can also show that it's dependent on the reaction conditions. So we can see that this is a useful way to look at the modifications, but even in a single protein, the amount of modification is different. This means that it depends on the accessibility of the individual cysteines to modification. If we go through, instead of just a single protein, a whole digest from a cell type here, rabbit heart reticulate, we will see many proteins, and some of them with multiple peptides, cysteine peptides, but for which the percent of cysteine oxidized is considerably different. So it allows us in a single experiment to identify all the proteins and to get quantitative information on each of the possible reaction sites. We've made another derivative recently to try to use the same way of reacting with cysteines, but in a much less expensive way and much easier to work with. And this is to take advantage of these fluorolabeled reagents, which can be bought, fluorinated acids and other things, which you can get and react them with specific molecules. In our case, we chose ones that would react with the cysteine. So if we take a BSA digest and a simple peptide with this fluorinated compound put in with it, we can see mostly BSA here. If we start eluting it with different compositions of the solvent, we fractionate the BSA, but not till we get to the really high organic solvent do we elute the peptide, which contains this fluorinated the fluorinated peptide. So we've changed just the physical and solvent properties of the peptide of interest, and this enables us to do the enrichment. So these are we're calling the fluorolabeled affinity capture. If we do MSMS on these peptides, then they behave exactly like the unmodified peptides, except that there's a mass shift at the position of the cysteine, which is modified. So this seems to be a very efficient and quite inexpensive way to get the same benefit as you could from the ICAT reagent. So in summary, the oxidative changes involve both level changes and post-translational modifications. Different sites in the same protein may be unequally modified. Multiple mass spectrometry and complementary approaches are useful to fully catalog these changes. The proteomic approaches may enable the subtyping of cardiovascular diseases and related diseases, and also the effect of different therapies, and to predict their outcomes. And what we can do then is, once we've seen these modifications, develop antibodies to the modified sites and use them for the routine analysis of these modifications. The ugly versions are, among those are the amyloidogenic proteins, where we have the proteins that have a normal functional state, and then when they unfold and refold into a fibril form, cause disease, which usually ultimately result in death. And two of the proteins we're working with the most are involved in systemic amyloid diseases. Transthyretin, which is a serum protein that's present at about 1/1000th the level of hemoglobin, though it has about the same molecular weight and is also a tetramer, and immunoglobulin light chains that are produced by an overproduction of a B-cell clone. And this leads to a sporadic systemic amyloid disease that is fatal unless treated, and only recently have there been any treatments available, and they involve stem cell transplants, and TTR, the only treatment available is a liver transplant. Well, this is the inherited disease. One of the ways for the identification of these diseases is to take a fat sample biopsy, and here's a comparison of a healthy patient and one with this inherited TTR disease. You can see they look quite similar, but there are regions where there are extra peaks. And these are forms of TTR. This is the native form, this is the dimer, and these are degraded forms. And then other proteins like serum amyloid protein or TTR associated with ApoE. And even things like heat shock proteins and crystallins that change in their levels in patients with the onset of amyloid disease. So we're looking at some of the same ideas of here's the FTMS of human transthyretin. We immunoprecipitate that from the serum. Because its properties are so similar to hemoglobin, we have to get it separate from the hemoglobin. So we can immunoprecipitate it, and then by electrospray ionization, we can see and determine the molecular weight of the protein. And what we see is that each of these peaks is multiple peaks. Very little of it is the unmodified protein. Two of the major modified forms are cysteinylated, which is a well-known modification, but another one is S-sulfonation at the only cysteine, which is at cysteine 10. So we have S-SO3H. This is such a labile modification that until fairly recently it was totally unknown, but now we see it fairly frequently, and so it may be a widespread and probably important modification. In the case of TTR, what we can do is try doing top-down tandem mass spectrometry. If we do the nozzle skimmer dissociation, it results in complete loss of this sulfonation. If we do a collision in the quadrupole, again, complete loss. But if we do electron capture dissociation, there's absolutely no loss of the sulfate. So we can find its position, and doing multiple experiments, we can see that the data is very reproducible. So it says we can identify this protein, we can identify modifications in the sequence, and even this very labile modification we can find by the top-down sequencing. This is an interesting modification because we found there had been some ideas that in fact it may be stabilizing the tetramer, which would in fact be a good thing. And you can compare by fluorescence detected analytical centrifugation that if you just modify the TTR with a fluorescent label, here's the wild type TTR. If you modify it with the cysteine modification, in fact it destabilizes the molecule. But if you look at the S-sulfonated molecule, it's in fact been stabilized. So this, in this particular case, modification may be helpful. And one of the treatments that's being considered for this disease is to stabilize the tetramer. If you go to the unfolded form in fact may be toxic, more toxic than the deposit. So the idea is to maybe stabilize the tetramer. And one of the ways it's being tried right now is to take a small molecule called difluzinol and insert that, give it to the patient. It goes into the middle of the TTR. Here's the wild type again, and here's the difluzinol adducted TTR. So it is stabilizing it. And there are clinical trials ongoing now to see whether that may be a viable treatment for the disease. With the ECD, as I said, we get quite a bit of fragmentation. If we look in detail at various parts of the mass spectrum, then in the region where we would have the low end, here's the C5, C6, and C7 region, what we can see is that the fragment at the 5th residue is only one peak. If we look at where the 6th residue is, we have a pair of peaks that are 30 Daltons apart. And if we look at the 7th peak and everything else up after that, then again there is a shift, and this corresponds to the fact that the variant protein has the modification by exchange of the residue at position 6. So from this type of spectrum, we can get that very specific information. And we have both peaks because, of course, the patient, if they're a heterozygote, have both forms of the protein. We can get the sequence information from the C-terminus. Here's the ETD mass spectrum, or the ECD mass spectrum of the companion peak, the Y85. Again, quite a bit of sequence information. And here the ETD mass spectrum, which has similar fragmentation, but in the ETD we're going even farther toward the central part of the molecule, where often the ECD mass spectrum will get you some at both termini, but not necessarily in the middle if the protein is still somewhat folded. If you use an IR laser to activate it first, then you can unfold it and get more complete fragmentation. What you saw those lower peaks on the gel, and what we can see of samples like this is from a fibril deposit, a MALDI mass spectrum. In that region, a series of peaks from 8,000 up to 9,000, spaced by what turned out to be the distances of amino acid residues. If we select any one of these peaks and do MSMS, we get fragment ions that give us the complete Y-terminal sequence. So it says that in these, the Y-terminus is intact, that the truncation has been occurring about 40 residues in from the C-terminus. So again, relatively simple information, but this is in vivo fragmentation that's going on in the deposited one, but from which we can easily get that information from the mass spectrometry. So the in vivo circulating and deposited proteins are a complex mixture of native and post-translationally modified forms. The ECD and ETD improve our top-down capability. So far, the ECD seems to provide more sequence coverage, but the fragmentation efficiency in ETD is higher. But they're very sensitive to the tuning, and pre-activation with the laser should help to optimize these results. But it looks like it will give us a way to do fairly rapid screening of variants, and one that could be developed into a clinical type of assay. With the light chain amyloidosis, as I said, it's an overproduction from a B cell clone of the immunoglobulin light chain. And there are a lot of variations both in sequence and in post-translational modifications, and these lead to death. We look at these a lot by atomic force microscopy as well as mass spectrometry, because then we can look at the fibrils. And what you can see from a single patient is that the appearance of the deposits depends a lot on the environment in which they're deposited. If we look at the MALDI mass spectra of the samples taken from each of these, they look quite different. The light chain weighs about 23,000, but here the major peak in all of these is around 13,000. If we just look at the fibrils and do a tryptic digest, we'll see a pattern that looks similar from the different samples, and we can assign the major peaks tentatively to different parts of the sequence. We can do then tandem mass spectrometry, in this case MALDI on a Q-tuff, of these peaks and get enough fragmentation to be sure of the assignments. And what we can then see, though, is if we look at this sample from this fibril, is that we have peptides that correspond to parts of the sequence, but in this case the methionine has been oxidized. In this case, we have a peptide whose sequence corresponds to part of the light chain, but for a tryptic digest, it doesn't end at either lysine or arginine. So it corresponds to a version of the protein which has been modified in vivo before the digestion. And in fact, if we go through this whole sample, we see this methionine oxidation. We see some truncation of loss of this aspartic acid. And we see, I didn't show you all of them, but truncation at each of these positions. And then that mostly this whole piece of the protein is missing, and this corresponds to the weight of about 13,000. So the deposited form is mostly this variable region of the protein with various forms of truncation on it. And there's a glycosylation site here, and we can look at that. We see, here's some modifications that go on at the N-terminus, where we can assign most of the sequence, have some question in this region here, and we didn't have a gene sequence at the time this was done, but because we can get the masses with very high accuracy, we can assign the elemental composition even to the unknown peaks. And our guess was that this sequence might be what was going on, that we had two valines and then this glutamic acid, and that this underwent oxidative deamination to alpha-ketoglutaric acid. So a lot of chemistry going on there. But really there's much more chemistry going on. And here we have another peptide from this light chain. And in this case, the sequence to be expected is asparagine. And this would have the molecular weight corresponding to this first isotope. The more abundant one corresponds to this being changed to aspartic acid. And it is true that slowly with aging, asparagines can go through a cyclic form and then reopen to aspartic acid. And this is showing us that this conversion has occurred to some extent. And if we then look at the ECD mass spectrum of this peptide, we're able to distinguish for the aspartic acid, when it reopens, it can open either as alpha, the normal aspartic acid, or beta aspartic acid, which puts an extra methylene group into the backbone and may destabilize the protein and lead to some of the misbehavior of it and some of the problems that come with protein aging. So we can tell, because with ECD we can fragment the side chain, we can tell the difference between the alpha and beta aspartic acid and actually quantify the amount of each which is present. In this case, in another peptide, we see at the position here where we would just expect to have tyrosine, we have a peak that's 34 Daltons higher, and in fact a little peak 2 masses higher. And this corresponds to the insertion of chlorine into the tyrosine, which forms 3-chlorotyrosine, that occurs under the effect of myeloperoxidase. And here the S-sulfonation reaction has taken place, and we formed the S-sulfonate at the final position of the light chain, and then this wrapped around and interacted with the NH2 on an amine on the lysine at position 196, which we can find out by looking at this MS-MS spectrum, the MS-MS spectrum of that, and here's another case in which the modification has occurred but interacted with a cysteine. And so we form this type of a bridge. So all of these are intermolecular sulfonamides and thiosulfonates, and this chlorination reaction is also accounted for by the action of the enzyme myeloperoxidase, which is producing these reactive species. And some of this chemistry had been predicted by the Winterborn group, but it hadn't been seen in vivo. And these are all samples that were obtained, data that was obtained from patient samples without any particular enrichment to look for these. So they are occurring, and they would be overlooked in most proteomic studies, but they're very much present, and they can tell us a lot about the kind of stress of the system. And there's also glycosylation on these light chains, so we can look at the carbohydrate structures. There will be a variety of different glycan structures at each site. The temptation is just to assign them from their molecular weights, but really you need to look at their tandem mass spectra in order to look in detail at the structures. If you then select any of those peaks, you can fragment them so that you get the sequence of the sugars by cleavage at the glycosidic bonds. But if you get fragments that go across the ring like this one here, that will tell you how much the weight is at the modification from that position. And if you get a fragment with the modification at a different site on the sugar ring, the mass shift will be there. So it gives you information not only about which sugar it's modified on, but which carbon atom within the sugar the modification has occurred. So for these, the proteomics provides a novel diagnostic tool and gives information on tissue damage, including oxidative stress. And the comparison of these fat tissue maps gives us information on up and down regulated and associated proteins. The glycomics can help us to correlate the glycan structures with the mechanisms for amyloidogenesis. And what we've noticed over a period of time is that in the variant chains and the sequence changes, that changes in the amino acid sequence that create new glycosylation sites are overrepresented in the proteins which are more strongly amyloidogenic. And the fat tissue biopsies help us to characterize the amyloid proteins and their fragments with the different systemic amyloidoses. But to understand the fibril growth and the modifications, this will help us learn about the circulating amyloid proteins and also their deposits and get at the mechanism of these diseases. And finally, an amyloid or a misfolding one, which provides actually some good, but really not good enough cell defense against the misfolded proteins, one in which we go from the native protein to an abnormal one, which can refold in a good form or can be degraded, or if it makes aggregates, can form toxicity. And what we're looking at here is Huntington's disease as a system and using yeast as a model. And what we found is that there's a microtubule-dependent transport of these misfolded proteins to form what's called an agrosome. And the formation of these can actually lead to survival because it locates the protein in a way which gets it out of harm's way. And if it's degraded, then it leads to autophagy. So as the model system here, we're looking at Huntington protein with different length extensions of the poly-Q. And the longer this is, the more earlier onset and more severe the disease is. So we have these long poly-Q sequences. You notice there's also a proline sequence. And what we've looked at in the model is varying these structures and finding out how much aggregation occurs and what other proteins are coming down with it. Because they're very insoluble, we had to go to methods like small continuous flow electrophoresis, as well as 2D electrophoresis, and then LC-MS for the digested proteins. We can see quite different patterns depending on whether we have the ones here with the proline region or without. And we can identify from the LC-MS different proteins that are associated. In this particular case, the 1433 protein, but there's two very closely related forms labeled 1 and 2, which differ by only one amino acid. And this single peptide that's circled there is the peptide that has the potential to tell the difference. If we do MS on that, it tells us that it's this sequence and not the other because the rest are the same. So it tells us we're dealing with this protein. What we've done is gone back through and then delete almost every one of the ones that seem to be involved and see what effect this has on the deposition process. So we identify these proteins and then we go back and do molecular biology to make constructs that leave out one after the other. And we also then look at these proteins to see what other modifications are going on. Although there's evidence of ubiquitination in the deposit, in fact, the Huntington protein itself turns out not to be ubiquitinated under these conditions. And so by these various steps, we can make a model of a pathway of what proteins are involved and what the structural requirements are. And we see that both a certain length poly-Q region and the proline-rich region are required for this targeting, for the assembly with these other proteins. All of the ones marked here are essential to cause this microtubular transport to form the agrosome. And now we're looking at mammalian cells to see if this same process is the same. So this agrosome formation, it does occur in yeast, so it can serve as a model. And the proline-rich region is the agrosome targeting region. The N17, the first region of the N-terminus, and the P region are critical for this targeting. And then these other proteins were shown to be critical by these deletion studies. And this targeting relieves the toxicity, so there's some genetic evidence for the protective role of these agrosomes. So in conclusion, the full characterization of these proteins and the understanding of the functions related to the diseases demands thorough analysis of the many different forms of the proteins which occur in the in vivo sources. The different types of mass spectrometry and tandem mass spectrometry allow reliable profiling, but also characterization of the post-translational modifications, but gives us consideration also of other species that may be involved. And I didn't have time to talk about them here, but this includes, very importantly, lipids and glycoconjugates. And the new mass spectrometry and separation methods are enabling these. But complementary, and I tried to show you at various stages, both chemical and other instrumental and molecular biology methods should be incorporated into these strategies. Finally, I want to thank the many people in our center whose work has provided these results, particularly my joint faculty, Josiah, Mark McComb, and Cheng Lin, and the many students and postdoctoral fellows in the laboratory, and particularly NIH for their generous support from NCRR, not only for our resource, but for our instruments, including the Orbitrap and the new high-performance FTMS, and our MALDI-TOF instrument, and NHLBI for our cardiovascular proteomics center, and our collaborators funding for the amyloid and Huntington studies, and in particular the BU amyloid treatment and research program, which goes from ugly guys to, in fact, friendly guys, we hope, in the long run. And I thank you all once again for the invitation and for your presence here. And if there's some questions, I'd be happy to answer them. Otherwise, we see you at the reception. Oh, sure.
A question. You have a reproducible way of looking at those changes of proteins. in the pathological process or other changes that occurs during oxidative stress. So how about the intervention? Because a lot of the antioxidant effects of different types of vitamins and other things has not shown to be effective. So you think by adding those supplements, you could also see if there is a real alteration or it is of no.
Well, what we can at least do is set up ways here to see what the effect of each one of these possible treatments would be to anticipate which may be effective, such as we did with that simple nitrite study. We can see for what is usually regarded as a very mild drug that in fact there's quite marked changes that go on in a lot of different proteins that last for quite some time. If we can use the same sort of approach for other potential treatments, see which systems are being affected, then that would help us to select what's most appropriate. And in other cases for individual patients, we have another study going on for which we had published some results in analytical biochemistry just a couple of months ago by looking at patterns of small molecules after treatment with a drug by the Huntington patients versus controls. We can see different pathways which are affected, some in the rate of the metabolism and direction of metabolism of the drug itself, but also we saw changes in tryptophan-related pathways that were particular to the Huntington patients. So in some cases you see the things you're looking for, and in others, because it's unbiased, you may find out quite a bit about a class of things that you hadn't known you should consider. Though the most effective proteomics and glycomics are usually when you have some idea and you do a more targeted study and not just try to look at everything everywhere.
In terms of the nitration of ischemia reperfusion injury model, how long did you follow it? Does some of the changes reverses at all, or they build up?
This showed you results out to 48 hours. We looked at a few samples past that, but there didn't seem to be many differences after that. As far as some of the changes, if they were a week later, we didn't look that far out, but we were surprised that our reviewers were surprised that we saw things even that lasted for 24 hours. So a follow-up would be to go back and look at some of these things with much longer term, especially if you make some metabolites that have the possibility for interaction with proteins that could form, like in the case of the lipids, which are oxidized, that then make condensation products with the proteins. Those would be irreversible modifications that would persist for whatever is the turnover time of the protein.
And just as a reminder, there is a reception in the library immediately following Dr. Casella's lecture. So please join us.
Thank you.